Welcome, dear viewers of the Vijay's Classroom of Biology. In today's class, we are going to see in detail about the ELISPOT assay. So, it is a sensitive technique that allows enumeration of individual immune cells capable of secreting specific proteins such as antibodies or cytokines with a very high sensitivity and at the single cell level resolution. So these are all the steps involved. First, this ELISPOT assay combines both principles of cell culture technique as well as the ELISA together in order to identify and enumerate the immune cells capable of secreting proteins from a population. So first is the cell preparation. Isolate and purify the immune cells of interest to be analyzed in the ELISPOT assay. And secondly, we coat the plate with specific capture antibody to capture the secreted proteins of interest. Then we add the immune cells to multiply, and we add the antigens in order to go for in vitro stimulation. We stimulate the immune cells with specific antigen or mitogen to produce specific antibody or cytokines of interest. And the fourth is the deduction assay where a spot will be developed. So we add biotinylated antibodies specific for the another epitope of the secreted antibody or cytokine from the immune cell. So these biotinylated antibodies can interact with the streptobidine labeled enzymes. So after addition of the enzyme, we add substrates. So maybe tetramethylbenzidine, or sometimes if it is horseradish peroxidase, we can add tetramethylbenzidine. If it is alkaline phosphatase, then we add phosphorylated substrates. So cleavage of this substrate in the presence of enzyme labeled antibody creates visual spots on the individual cytokine secreting cells. So in this way we can identify the number of cytokine secreting cells or cells that are capable of secreting specific antibodies of interest among the population of immune cells that we have collected from an individual. So there are very many applications. First is cancer immunotherapy. Here we assess the cytokine response of immune cells to evaluate the efficacy of immunotherapy treatments. And secondly, in vaccine development, we evaluate the vaccine induced immune responses just by quantifying the antigen specific cytokine secreting cells or antibody secreting cells. And in case of autoimmune diseases, we can investigate the role of cytokines in autoimmune diseases that will help in diagnosis as well as therapeutic options. Advantages, there are three major advantages. It is highly sensitive technique. It can detect even low frequency of cytokine secreting cells in a population. And single cell resolution. It allows the analysis at the single cell level. We can identify and observe individual cells capable of secreting specific cytokine and antibody of interest, providing a valuable information about the heterogeneity. Though there are heterogenic population of immune cells we have collected, we can still identify the number of cells secreting independent cytokines independently and antibodies. It is a quantitative measure. As I told you, we can count the spot, we can enumerate the spot and bring quantitative data allowing a direct comparison between the samples in a population. That helps in research study also. There are some limitations. Variability. So results can be influenced by factors like cell viability, enzymatic activity, and antibody specificity. So we have to use the highly specific secondary antibody to identify, and there may be non-stop enzymatic activity. We have to stop the action in order to detect it. But anyway, sometimes enzymatic activity and maybe the immune cells are dead, but still the secreted cytokines may be present in the cells creating spots. So it's a matter to consider and these may introduce some variability in the results in the final data sets. And there can be cell subset identification. So ELISPOT identifies cytokine secreting cells but may not differentiate specific cell subtypes or subsets within a population. So whatever the interleukin, let's say for example interleukin or interferon, gamma secreting cells, whatever the cells are there, they are secreting those cytokines may be detected, but there may be a case that some of these immune cells might secrete these cytokines because of some other antigen with which the individual has exposed. So not exactly it's going to be for our antigen of interest every time. Subjectivity in spot counting. So manual counting of the spot can introduce subjectivity and variations between observers. So as we visualize the spots and manually count, there can be some variations or errors when skilled individuals are not there. That may be chances for error to occur. So conclusion, so it is a versatile and powerful tool for immunological research, providing valuable insights into the cytokine based immune response as well as cellular immune function at a single cell level. Thank you.